and welcome to our second interview in the Oxford Brookes Biomedical Society podcast series. With us today, we have Dr. Jonathan Lees, a senior lecturer in bioinformatics. His journey began at Durham University, studying molecular biology and biochemistry, and then a research master's at the University of York in bioinformatics. But that was not before taking out a year to cycle from the UK all the way to China. I personally want to talk about this more because that's just crazy to me. But then when he came back to the UK, Dr. Lees undertook a PhD at Birkbeck College London under Dr. Wallace in crystallography. And then combining his work in bioinformatics and crystal structure, his postdoctoral research at Queen Mary was on machine learning approaches for spectroscopy analysis. After that, he continued on to become a postdoctoral research fellow at UCL, where he worked on high-throughput protein sequence function and the structural annotation of data. One thing that I would love to highlight about your time at UCL was your work as part of the Oregano group on the website CathGene3D. For those unfamiliar with Cath, it is a protein structure classification database widely used in the bioinformatics field to find the evolutionary relationships of protein domains. All of this brings us to 2018, when Dr. Lees joined us at Oxford Brookes as a senior lecturer in bioinformatics. One of your most notable accomplishments is coming joint first in the prestigious CAFA Protein Function Prediction Challenge with your novel machine learning method. Hi, Dr. Lees. Thank you for joining us today. That is an incredible resume. So, yeah.
Yeah, good to see everyone. Yeah, thanks for that introduction. Yeah, it's been a bit of a journey for me. But yeah, thanks for the opportunity to talk today. So it's a long time to finally arrive at Oxford Brookes, but it's been a great time. So please ask any questions.
So one of the first questions I had was your year out before your master's, cycling from the UK to China. That's I googled it. That was 4,831 miles. How did you do that? The resilience.
Yeah, a friend of mine, we used to always go mountain biking, and we'd done little trips to Romania and the Alps, and sort of, we'd go places most weekends. And then he just phoned up and said, "Oh, do you fancy cycling to China?" And I was like, "All right, yeah, fine, yeah." So it was kind of mountain biking really. So we did lots of off-road routes over mountains and things, and we sort of took a funny route, but it was very exciting. And I'd recommend to people, it's good to take that year out whilst you can. For me personally, it was a really, exciting time, and I look back very fondly on it. A lot of stories from that trip.
Gosh, I hope you didn't get too lost. I can't believe maybe using Google Maps or, while cycling, a little bit tricky.
That's right. It's kind of before the, we had like phones with, you could tell where you were. So we had, I think at one point we were in like the Tibetan plateau.
Oh my gosh.
Had like a tiny map about the size of an A4 piece of paper. We decided whether or not to go left or right. And a couple of days later, we just had to go back because we were just in the middle of a giant mountain range.
Oh my gosh.
By, rivers.
Yeah, but the scenery must have been amazing. Like what, like a sight to see. I mean, gosh.
Yeah, it really was. Yeah, mountain biking is a great way of getting around and seeing all these things. You sort of really take in it's really nice to slowly see the change as you go further and further. You see how everything's sort of just sort of gradual and continual, and a lot of nice food and on the way and everything. So really good. Yeah.
Fantastic. And so it sounds like really an adventure. Pardon?
That's one of the reasons I got into bioinformatics, is because the cycling kind of knackered one of my knees. So I needed to find a job. I'd like to think of a better reason, but I kind of got into it because I needed a job where I could just sort of sit down and program a little bit.
So you could have become maybe a professional cyclist, maybe, into
it was never that good, but we got pretty healthy. It's quite a surprising.
I was going to say, keep you fit. So speaking about you entering into the field of bioinformatics for, an unfortunate reason, but I think it turned out all for the best, everything happens for a reason. And the sphere, the entirety of bioinformatics is such a big field, and not a lot of people are completely familiar with it or understand it properly. So in a few sentences, can you give us a better idea and a professional explanation on what bioinformatics is really about?
Yeah, so yeah, you're right. It's kind of a massive area now. Probably there's lots of different summaries you could do because bioinformatics is many things. Probably the most concise definition is something like taking these large data sets that you get from various projects now, the Human Genome Project being one of those, and trying to derive knowledge from those data sets. So it's kind of taking this raw data and trying to extract meaningful knowledge that we can then go and test experiments and things like that with. So but there are many aspects to it. So other aspects are obviously developing algorithms, things like BLAST, one of the first bioinformatics kind of innovations, that's one of the most cited papers ever. Also that's, all the databases and the resources. And then, but I mean, in some ways, biology, as you probably see yourselves going through your degree, it's kind of turning into a bit of a data science slowly. So as a lot of the more simple questions are kind of, or the low hanging fruit is kind of tackled, we need more and bigger data sets to kind of ask more complex questions. And then bioinformatics is kind of coming into, in the field of cancer, which we can maybe talk about a bit later, to really kind of understand these complex biological systems. And so inevitably you need computers to kind of analyze that data. So yeah, but I think the simplest sort of definition is just taking that raw data and trying to extract knowledge out of it. That's kind of one definition that you could use.
It's interesting how you said that a subject like biology, which is really kind of known as a textbook kind of subject, is now almost becoming a data-based one. Like, who would have thought now in these recent years the importance of computer tools or instruments have such an influential role? And like you said, especially with cancer research and now going down the route of personalized medicine and genetics, it's such an important now frontier. I think there's a lot of investment, so we need more people like Dr. John Lee's, on it and helping you discoveries.
Yeah, there's definitely a big future for people. So I'd encourage people to get into it, if that's the sort of thing you like. And yeah, definitely, I think with the Human Genome Project, that's one of the things that really kickstarted the whole field. It was around before in kind of structural bioinformatics in a big way. But yeah, I think these big data sets, these big projects like the Human Genome Project have really kind of, that's really what kind of got the ball rolling a lot faster.
Yeah, and funnily enough, we actually spoke to Dr. Dave Carter last week, and what he did in the Human Genome Project. So it's interesting. I mean, he was only a PhD student at the time, but I think it's such. I don't think we realize how much of, I guess, domino effect it will have for the future. Such an immense project. I mean, who knows what extent these things now using the Human Genome Project can go to.
Exactly. So yeah, I think things like, that's leading now to personalized medicine. So in the field of cancer biology, where you can sequence an individual's tumor, you see what's particular to that tumor, and you can then start to derive, treatments that should work for that individual. So we're really just at the start of that. But I think fields like that are really going to be where, the public become a lot more aware of bioinformatics as it kind of starts to come into the clinic a lot more. So people, in the field of cancer, they'll have the cancer sequenced, and then there'll be some bioinformatics people and algorithms kind of, analyzing their data. So I think it's kind of moving into the mainstream more in the near, next 10 years.
No, but it's amazing. Like, I mean, I don't know how advanced the personalized medicine frontier is at the moment. I mean, it's still very much up in the air because I don't know if it's an ethical issue or just a practical issue. I don't know how much it costs really to try and analyze someone's, like, a tumor's genome. I don't know the costs behind it. I mean,
yes, I mean, it's really sort of, the cost is coming down very rapidly. So in the future, that's not going to be so much of an issue. It's really kind of, trying to being able to read this, read the book of life, read the genome. So that's kind of the issue, and that's where a lot of the technical developments are at the moment. So it's just it takes time for all of these things to come through to fruition. But yeah, there's a classic plot on the cost of sequencing a genome, and it comes, eventually it's going to be, $10, and then probably one day it'll be $1 to sequence a genome.
Crazy.
Because it just gets cheaper and cheaper. It's just getting cheaper and cheaper.
Yeah, I think we were doing what was it last semester? It was the genetics module. And I think in one of the lectures, it mentioned about like the cost about, I think it was sequencing can't remember which organism it was, but it was in the millions. And this was early days in sequencing. And now if you think about it, it's under maybe £100 or something, like going from millions. I mean, it's mental.
It just lends itself well to being sequenced. And then the challenge is not necessarily getting the sequence data, it's interpreting the sequence changes and all of the downstream effects. That's where the complexity is. And that problem is so complex, we'll all be in jobs for hundreds of years analyzing this, because it's not necessarily like the field of physics where it's kind of, it's very clean and straightforward. Biology is really kind of
it's unpredictable. I think that's almost the beauty of it, trying to find order in something so chaotic. It's a bit of a challenge, isn't it?
So as a lecturer at Brookes, I know that you run the research methods module and also ran a coding club at one point, but as a researcher, you are the PI of Lee's lab, which looks at three main areas: structure and function of proteins, disease informatics, and machine learning. Can you tell us about how these three fit together?
Yeah, sure. Yeah, so I mean, my background really comes from the field of taking protein sequences. So as we just mentioned, there's genome sequencing getting a lot cheaper. So we've now got hundreds of thousands of genomes already. Soon we're going to have millions of genomes. Eventually, who knows where it's going to end. But all of those genomes, we can extract the protein sequences out from them. Reasonably straightforward. It's quite straightforward. But then trying to understand what those proteins do in terms of their function, what they look like, so in terms of their 3D structure, and starting to understand, how the proteins fit together with other proteins. That's the sort of thing that the amount of experimental annotation in these data sets on the protein sequences is really quite low still. So even though we've got hundreds of millions of protein sequences, we've only got 100,000 or so experimental annotations. So this is sometimes called the functional annotation gap. And that gap is just going to get bigger and bigger over the next 10 years. So for example, the Sanger, which is a world leader in sequencing genomes, they've announced very ambitious proposals to sequence the whole of eukaryotic life. So it's millions and millions of genomes, and they're doing really well at that. So really, this is like one of the key challenges: is how do we don't just want to leave these sequences sitting around in a database doing nothing. We want to mine them for novel functions, novel antibiotics, potential novel medicines, and even just to understand, the evolutionary processes, how functions evolve. It all ties together. So that's basically one of the focuses of that. So, and it stems from the work in the Orengo group. So we basically take protein sequences, and then we predict the structures of those on those sequences, specifically the domain structures. And we also annotate functions to those domains based on sequence patterns in the domains. So yeah, that's kind of the protein structure function prediction aspect. Now, sort of moving on to diseases, so obviously to take a human genome, even the human genome, which is one of the most well understood, well characterized genomes, there's still plenty of proteins we don't know their functions. People obviously know about p53, and they focus a lot of their research on those proteins, which is great. They are really important, but there's a lot more proteins out there that kind of are really important. So part of that is even the human genome, predicting the functions automatically from sequence is really important. And that's before you go on to diverse eukaryotes like malaria, trypanosomes, leishmania, all these really important disease organisms. So the ability to predict function from sequence is kind of one of the holy grails that's out there at the moment, and that's something we're working on. And it has obviously had relevance to disease, the two things kind of tying together. One area that I'm going into more and more is this field of cancer informatics that we discussed. So it's really, there's a lot more, like I said, there's a lot of really powerful data sets emerging. So one of them, just to mention briefly, is this CRISPR-Cas9 genome scale knockouts. So you kind of go through and knock out every single gene in the genome, and you can do that for multiple genomes. So at the Sanger, for example, they've got 400 models of cancer, and then they've knocked out each gene individually to see the effect on growth. Because cancers, they have a lot of strengths, they outcompete our own cells, but they also develop a lot of weaknesses and dependencies. We want to kind of try and identify and exploit those weaknesses through drug development. So what I'm doing with the undergraduates is we're looking at these large data sets. We're trying to extract knowledge, like I was saying. So we're looking for patterns in certain cancers that identify, they've got this dependency on this pathway. And what we've been finding out this year is a subset of cancers that really have quite altered metabolism, and they've got a much higher dependence on oxidative phosphorylation. And what we're doing now, in collaboration with other people at Oxford Brookes and Oxford University, is we're designing drugs. We're going to test drugs basically on some of these predictions. So that's kind of almost one of the frustrations of bioinformatics is sometimes you make predictions and you just, it doesn't go anywhere. But being somewhere like Oxford's great because there's a lot of it's very research intensive, so we can reach out to these other people close by and we can test these things. So it's been a really, hopefully, a fun project for the undergrads, and they're making a real contribution potentially. We don't know exactly where it's going, but that's one example of where we're using bioinformatics for studying disease.
With your undergraduate project, sorry, are you using CRISPR yourselves, like the Sanger Institute, or what other process are you doing to kind of extrapolate or trying to understand these data sets?
Yes, so that's the really nice thing about bioinformatics. I think it's almost the nicest thing is we can just go to the Sanger website, download this data. It takes a minute, and that experiment probably cost 30 million pounds or something to do. But
Oh my gosh.
we can download it and we can start to analyze it for free. The bioinformatics is really kind of you just need a computer, which isn't expensive nowadays. You can get computers online, even in your own browser. You can start to analyze these really expensive data sets. And what typically happens is, as these data sets get developed, they look for certain patterns, but they don't think of testing other things, or they don't think of combining it with other data sets, which is where you can get out, new knowledge and new. So there's a creative aspect to bioinformatics, which I really like, which is kind of thinking, actually, if we look at it from this angle, or if we combine it with this other data set and we take a slightly different approach, maybe we can find something else out. So it's a really kind of creative side to it that I think is probably my favorite part of it and the bit that I enjoy most. But yeah, it's really nice that we can go and test these in the lab now. And we've got some good collaborations there. So fingers crossed, we never know what we're going to find, but hopefully it's something that we could use in personalized medicine for saying, this drug will work on this subset of cancer patients.
It's amazing how just from, I guess, a process of elimination, or like you said, by interpreting the data, you can go so far as then selecting the types of drugs to use against those, I think, like those knockouts or those maybe overexpressed genes. Can you just give us a quick example of, I don't know how complex it is, like the process to then kind of decide, okay, X gene was investigated and then we chose Y drug to then act against it. I think that's something which is mind-boggling for me, how you can just go from one stage to another.
Yeah, so again, it's, what's nice nowadays is that all of this data is kind of well stored in databases, which is one of the aspects of bioinformatics. So just to give you an example back with that project, we identified, I think we found about 10 to 15% of cancers potentially have this dependency on this specific pathway in energy generation. Now, when we actually look at the genes involved, forms quite a tight set of genes all forming this generating energy for these cancers. And you can clearly see if any of those genes are kind of missing, the cancer doesn't grow well at all. So what we can see is there's a drug that's used in diabetes for, it's not used in cancer at all. It's used mainly in diabetes. And essentially, but we can see that it targets these genes. So our hypothesis that we generate then is that if we give this drug to specific subset of cancers, they're going to display, heightened sensitivity to this drug. So it shouldn't kill healthy cells. It probably won't kill all cancers, but it will. Our hypothesis and our hope is that it will be really effective against a subset of cancers. So it's kind of a bit of detective work. You kind of have to follow the trail of things, but again, we can find a lot of the information out from online resources.
Amazing.
I did have a question about that. About how far into the future do you think this will become applicable, or is it like something super close and we're just on the cusp of starting to use more personalized medicine using this kind of
I think we probably it's been kind of threatening to sort of happen quite some time, but I think it really is getting close now, especially in the field of cancer informatics, just because there's so many new data sets coming out there, like single cell data sets, where we can really see the heterogeneity in the cancers, and just also with machine learning and able to sort of look at these complex data sets, which we sort of talk about in a little bit, I suppose. I think a lot of things are coming together at the right time. The richness of the data, the computers, and so you're never quite sure when it's going to sort of clip. But hopefully in the next 10, 20 years, it's going to be a really exciting time, I think.
So too.